Good day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. We have discussed different algorithms for de novo genome assembly starting with next generation sequencing datasets. So, in the last class I introduced the last algorithm which was the De Bruijn graph assembly, and we talked about the steps that we need to perform to get to the reference sequence. So, what we will do today is we will look into special cases again with this De Bruijn graph assembly, and then we will talk about the subsequent steps after we have done the assembly. So, this will be the agenda for today's class. So, we will talk about DBG assembly with non-uniform coverage and sequencing errors, how this non-uniform coverage and sequencing errors actually affect the assembly process and what is the outcome. We will then talk about the different tools that are available for assembly. So, these tools are based on these algorithms that we talked about. So, I will just briefly mention those tools that you can use, and then we will talk about the assembly quality, right. So, once we have assembled the genome, we need to evaluate the quality of the assembly process, right. So, whether the assembly is actually good or whether there is some scope for improvement, those things can be assessed using this assembly quality process. So, I will discuss about that, and finally, we will talk about something called genome annotation. Once you have assembled the genome, you want to identify the different features that are within the genome. For example, you want to identify the genes or the coding sequences or exons, introns, etc. So, these will be the keywords for this class: assembly, quality and annotation. So, let us begin with the De Bruijn graph assembly. I will simply highlight the steps. So, the first step is to start with the reads that are of length k, and we generate the left k minus 1 mer and the right k minus 1 mer for every read that we have in the data. The second step is to generate this directed multigraph based on these left and right k minus 1 mers, because these left and right k minus 1 mers they share an overlap of length k minus 2. We utilize that property to generate the directed multigraph. In the third step, we actually look at the property of the graph that we have created, and we talked about these balanced and semi balanced nodes, and based on fulfillment of certain criteria, we can check whether there is an Eulerian path that is available in the graph, right. So, this is something that will establish, okay, there is an Eulerian path. So, we also talked about this Eulerian path, right. So, this is a path that traverses to every edge exactly once, and as we traverse through the graph using this Eulerian path, we actually generate the reference sequence, right. So, we derive the reference sequence by traversing through this Eulerian path. Okay. So, what will illustrate today is that we will see, right, De Bruijn graph assembly may not always keep the right reference sequence, and we will start with the same example that we talked about in the last class. So, why would this happen? Why would you not get the right reference sequence? So, because in a graph, right, that satisfy all the criteria, there might be multiple paths. Okay. It is not necessary that you will have just one path which is unique, but you can have multiple Eulerian paths, and we will see in an example that these multiple paths can give rise to different reference sequences, right. So, again these are highlighting the limitations of the assembly algorithms, right, and it is good to know this, because then we can think about ways to actually address these issues. Okay. So, let us start with the same example that we actually worked with in the last class. Okay. So, here is the reference sequence on top. You can see the reference sequence is the same reference sequence that we talked about in the last class. Then we have reads of length 4. Okay. So, here the k equals to 4, right, and I have written down all these 4 mers for this reference sequence. Okay. So, what we do now is from these k mers we generate the left and right k minus 1 mers, right. So, as we discussed, this is the first step. So, that is what we do, right, and from that k minus 1 mer we generate this directed multigraph. Okay. We generated this directed multigraph in the last class. So, I am not going to go into that steps again. Okay. So, what I am showing is the directed multigraph with the nodes. Okay. So, these nodes are k minus 1 mers, the connections, these are the edges, right, showing the overlaps, and they actually signify the k mers. So, as we traverse through an edge, we get a k-mar. In addition, I am showing these two numbers which we also introduced in the last class. So, the first number is the number of incoming edges, right. So, this is the in degree for a node, and the second number in blue, that is the number of outgoing edges or the out degree for every node, right. So, we are using this convention to denote this in degree and out degree for this node. Now, why are we considering this in degree and out degree? Because this is important to decide or to derive whether there is an Eulerian path available in this graph. Okay. And the condition was that there could be at most 2 semi balanced nodes, okay, and the rest of the nodes should be balanced. Again, reminding you about balanced and semi balanced nodes. So, balanced nodes are the nodes where the in degree equals the out degree. Okay. So, in this graph what you see, all these nodes here, right, these are balanced nodes. Okay. So, the in degree and out degree, they are identical, but then you have two other nodes, okay, this one here and this one here, where the in degree and out degree are not the same, but then the absolute difference between in degree and out degree is equal to 1. Okay. So, this means these two nodes are semi balanced nodes. Okay. So, this graph then satisfy the criteria that we defined, right. So, you have at most 2 semi balanced nodes. So, we have 2 here and rest of the nodes are balanced. So, which means there must be an Eulerian path in this graph. Okay. So, this is the condition, right, which is satisfied. Now, we have to find the Eulerian path. Okay. And last time we traversed through this graph to actually find the reference sequence, and I just mentioned, right, we have option, but we took one path and we actually got the right reference sequence. So, if you go back to the last class, you will see, like, this is the path we took, right, which I will highlight with these red arrows. As you can see, we are traversing between these two nodes GGC to GCA, and we got this GGCA, right. So, this is the reference sequence that we are building as we are traversing through this path, right. So, remember, every edge actually gives back the k-mar. So, we took this path last time, right. So, we are going in this direction from GCA to CAT with this red arrow, then from CAT to ATG, right, and as we go along, we are also building the reference sequence at the bottom. You can see that I am just adding the letters, and then we completed this process. Again, without going into the full details, we completed this process, right, and we actually traversed through the full graph, right, and then finally we reached here, and that is the end, okay, and we got back the right reference sequence. Okay. So, this is something we traversed in the last class. Now, the question is, are there alternate paths in the graph? Okay. We start at the same point, right, with that red arrow, right. So, we start at the same point. Is there any other path that is also Eulerian? Okay. So, let's look at these options, right. So, here you have these two options. So, last time we took this option, right, here we took this branch, but now we also have this branch. So, what happens if we traverse through this branch, right, which is highlighted by this blue color, right. So, blue color arrow you can see. So, if we traverse through that branch, what happens? Okay. So, let us traverse through that, okay, and let us find let us see whether there is an Eulerian path, and if there is, then let us find the reference sequence for that path. Okay. So, we traverse through this. Now, I will show you step by step, again now highlighting with blue arrows, okay, and this is the path that we are taking. We are also building the reference sequence, and then we take the self edge, right, we are adding these letters to the reference sequence, then we take the next step, then the next one, right, we are going through this, right, these are the solutions. From this ATG, now we have to take this to TGG, right, that is the way the arrows are given, right, in this graph. So, we go to TGG and we then go back to this GGC, right. So, that is where we started, right. So, we have now again this option GGC to GCA, there are two edges, right. So, we are taking the second edge now, we are traversing to the second edge. In essence, we are kind of now utilizing all the paths that are available, or traversing through all the paths that are available. Now, from this point, now we can take the upper path, right. So, towards CAT, right, because we have taken the downward path already. So, we take the upper path CAT and then we reach the final node ATG. Okay. So, we have traversed every edge exactly once in this graph, and that means this is an Eulerian path, okay, and corresponding to that path we have got the reference sequence. Now, we can compare this to the reference sequence that we started with, okay, and it turns out this is not the correct reference sequence. Okay, and I have written down the correct reference sequence that we got earlier, and you can see this difference, right. There is this difference. So, first two or first three actually they are identical, and maybe first four are identical, right, and then the last three are also identical, but then in between there are some changes. Okay, and you can see the order is different. This AAA TGGC, this order is different between these two reference sequences that we derived from the same graph. Okay, and that is because of the specific structures. Maybe some elements are repeated. So, that is where we are getting this kind of structures where you can have multiple Eulerian paths, and we end up with different reference sequences. Okay, so, this is something that we should keep in mind, right. It is not always necessary we will have a unique reference sequence derived from a multi-graph. Okay. Okay, so, now what we will do is we will take another example where we will actually do the assembly with repeat sequences, okay, and we will see how de Bruijn graph actually handles them. Okay, so, we will take this input sequence. We have used this input sequence before for VLC approach, etc. Right. So, we will take this example of very warm day. So, we see this repeat very clearly. You have this very part, right. So, this word is repeated 3 times in this sequence. So, this is the repeat region, right, and let us build the de Bruijn graph. Okay. So, we work with reach sequence of length 6 just as an example. You can try with other lengths as well, that is not a issue. So, the process is exactly same. Okay. So, here are the reads that you can find from this sequence, okay, and you will probably notice some of the reads are repeated, right. So, because they are coming from the repeat regions, and one of the assumptions that we are making is that every region is represented equally. What it means in other words is that we are expecting uniform coverage. Okay. So, every region of the genome is covered uniformly. So, we expect uniform coverage, and we will see later on what happens if we deviate from that assumption. Okay. So, now with this ideal scenario, right, we now build all these left and right k minus 1 merges. Okay. Of course, I will not have time to describe all this. So, you can see them on your screen. You can take a pen and paper, and then you can derive all these left and right k minus 1 merges yourself, right, following the same process that we described before. Okay. So, once we have built this left and right k minus 1 merges, we can now build the directed multi-graph. Okay, and this is how it looks like. So, again the process is exactly same as before what we have discussed. Okay. We add these edges between left k minus 1 mer and right k minus 1 mer, and we kind of keep on adding edges, right, and we end up with these multi edges, and until we exhaust all the reads that we have. Okay. So, we follow the same steps now, right. We can identify what are the balanced nodes or semi balanced node by pointing out the in degree and out degree values for every node in the graph. Okay. And as you can probably see in this figure, right, you have 2 semi balanced nodes, right. So, here is one, right, this is semi balanced, right, and here is another one. Okay. So, these 2 are semi balanced because the in degree and out degree, the absolute difference is 1. For the rest of them, they are all balanced because in degree is equal to out degree. Okay. So, we have 2 semi balanced nodes, and which means there is an Eulerian path in this graph. Okay. And what we have to do is then traverse through this graph through this Eulerian path and derive the reference sequence. Okay. So, let us do that, right. So, we start from this point, right. Again we start with from an edge which is semi balanced and where the out degree is greater than in degree, right. So, we start from here, okay, and we are traversing. Again I am highlighting these paths through this red color, right, red arrows, and we kind of I will kind of showing this very simple step because this is kind of we have straightforward paths, right. So, we traverse up to this point. Now, we go up, right. So, we are taking this path here, we are going up here up to this point again, okay, and then we traverse to another edge, right. So, another round of traversal through this region, right, through these nodes, and then we go up again, right, using the black ones. So, black ones we have not traversed yet, right, and then we come back, come down here to this node, right, and now we have to take the right, right. We have completed traversal of this part. So, we can take the right now, and once we take the right, you can see this is very straightforward, right. We have this simple path that can traverse through all these nodes, and we end up with the reference sequence on the right. So, as we are traversing, you will see I am writing this reference sequence on the right, and we are getting the reference sequences derived as we proceed through this graph. So, what you notice in this case, right? So, we have derived the reference sequence including the repeat sequences, right. So, this is the correct reference sequence with the repeats that are available. So, what you can see, at least from this ideal example, that ideal scenario, right, DBG assembly can resolve repeats to some extent, but then the assumption is that all regions are covered uniformly by the reads. Okay. So, and this is a very critical assumption, right. The moment we shift from that, the whole thing will change. Okay. Now, why I am saying this is critical, because in real life it is very unlikely that you will get uniform coverage from the from all the regions in the genome. Okay. And in that case, you might not be able to find Eulerian paths, or the graph may not be Eulerian. Okay. So, what will happen if we have non-uniform coverage, right? And we will see, like, this can lead to graphs that will not have any Eulerian path. Okay, so this is the example now we will take. Okay, we'll do the assembly with non-uniform coverage. Okay, and we take the same example. What I will do is, so I have also written down the read sequences again as before of length 6. To generate this non-uniform coverage, what I will do is, like, I will remove one read from the data. So, which I have highlighted here, you can see, right, this read is removed. I have removed this read, right. So, this means this will create this non-uniform coverage, because a part of this reference sequence will not be covered by the reads. Okay. So, what happens now? So, we go through the same process. I have remove this k minus 1 mark construction for this read, because this read is not present anymore, right, and we can use then rest of the k minus 1 marks to create the graph, multi-graph that we created as before. Okay. And once you create this, right, so what you will notice is from this one, right, this connection, this one of the connections will be removed, right, because this read is not there anymore. So, these two k minus 1 marks will not be there in the graph, and which means we will have one less edge here between these two nodes. Okay. Now, the moment you have this difference, right, this read is removed, what you see is that we have created two other nodes that are also not balanced. Okay. So, we have had two nodes that were semi balanced, right, were not balanced, but we have now created two extra nodes that are not balanced either, okay, because of this removal of this edge. Okay. Now, you can see the in degree is not equal to out degree, right, they are semi balanced, the difference is 1, absolute difference is 1. Okay. So, which means in this connected graph we have 4 semi balanced nodes, okay, and this means we cannot say that there is an Eulerian path, right. So, there is no Eulerian path, because we are not satisfying the criteria that we discussed before, right. So, for that for an Eulerian path to exist, the graph should have at most 2 semi balanced nodes, but here we see 4 semi balanced nodes. Okay. So, this is the problem now. So, if you do not have an Eulerian path, how do you define how do you derive the reference sequence, right. So, that's the problem that we end up with, and you can probably see now is we will not be able to derive the reference sequence. Maybe some part of the graph will be Eulerian, maybe you can have you can disconnect certain parts, you can probably see, right. Maybe this right part here, this part, if you remove this part, right, this edge, and you can see this part of the graph will be Eulerian. So, you can derive the reference sequence. Okay. And similarly, you can see whether certain parts of this graph are Eulerian, and maybe you end up assembling fragments of the reference genome. Okay. So, these fragments are the contigs that we discussed before, right. So, this is what we usually get at the end of the sequencing process, right, at the end of the assembly, because of these reasons, right, because you will not get equal coverage or uniform coverage across all regions in real sequencing experiments. Okay. So, we will talk about another scenario. So, we have to do assembly with sequencing errors, right. So, this is quite common, because the sequencers that we work with, they make errors. If you are working with short reads, there will be errors. If you are working with long read data, nanopore, etc., you will also have error, and also probably a lot more error, right. So, this is something that we would have to deal with the real world, where we cannot just apply the de Bruijn graph assembly in the ideal world, right. So, we have to deal with these issues in the real world. So, let us see what happens if we have a sequencing error in one of the reads. Okay. So, for doing that, we have taken the same example, okay, with the same reads. What I have done here for this one read here, I have changed, okay, the letter, okay. Instead of this R y, I have now made it R z. Okay. So, this is a sequencing error. This Z did not exist in the reference sequence, and we have got a different letter here. So, let us see how this will impact the de Bruijn graph assembly process. Okay. So, again as before, we are getting the same k minus 1 merge. Now, here is the read with the mutation, right. We have this R z underscore V E R, right, and we have derived the k minus 1 merge left and the right ones. Okay. So, what we are going to do with this is now construct the directed multigraph. When you construct the directed multigraph, what you will notice is that this R z underscore V, this does not exist, this k minus 1 merge does not exist in the graph, right. So, what will happen is that these two k minus 1 merge, they will form a separate graph, okay, which is disconnected from the main graph. And on top of that, what you will notice is that we have again created two nodes that are not balanced. Okay. So, we already had two earlier, right, one at the beginning, one at the end, but in addition we have created two other nodes that are not balanced. Okay. So, again you cannot find an Eulerian path in this directed multigraph. Okay. As before, you can probably disconnect certain parts and see if that part of the graph is Eulerian, and perhaps you can derive the reference sequence. And on top of that, you have this disconnected component, right. So, this is something again we cannot do anything about, right, and we might then have to simply just keep this disconnected component, we cannot really derive anything. Okay. So, as you can see, this sequencing errors can again disrupt this assembly process. Okay. And as I mentioned, this the biggest component is not Eulerian because of these reasons that we just discussed. Okay. So, one of the other points that I just wanted to mention is that in de Bruijn graph, we also sometimes use edge weights. Okay. So, this is something that we do because if we have, let us say, a average coverage of 30, 50 or 100x for a genome, right, you do not want to have these 100 edges tracked, right. So, what we do, instead of drawing all these edges or storing this information, we can simply put a number saying there are 100 edges, right. So, those connections, number of arrows or number of edges could be the weights of those edges, right. So, I will just illustrate, right. So, this is actually done for coverage where you have this high coverage for genomic regions, right. As I mentioned, it can be 50 to 100x, and instead of drawing or keeping track of all edges, right, when you are doing the computation, you can give one edge weight for each distinct k-mer. Okay. So, this k-mer is the edge, right. So, an edge represents a k-mer that we have discussed. Okay. So, I will just illustrate with this example, right. So, this is the same example as before, we have these edges. Now, here the numbers are small, but you can imagine if you have, instead of these 3 or 4 or 2, right, you have let us say 30. Okay. So, we have to keep track of all this information. It is better actually than to actually convert them to number and say we have 3 here, this will automatically signify we have 3 edges here. Okay. So, this is something that can be done. Okay. So, to summarize what we have discussed about de Bruijn graph assembly. So, DBG assembly does not require pairwise comparison of reads, right. This actually saves time, okay, and this means this algorithm will be faster than the other methods that we discussed, namely OLC or the SCS. So, what we have also seen, if a graph satisfies this criteria for Eulerian path, we can get the reference sequence by traversing through this path in the multigraph. And we have also seen there are different scenarios where you can have multiple Eulerian paths and we might end up with a wrong reference sequence. And finally, we have also seen that if you have non-uniform coverage, repeats and sequencing errors, these can affect DBG assembly, right. So, we took some illustrations and we have shown like this can disrupt the assembly process, and of course, there are ways we can address this. One is of course look for subgraphs that can be Eulerian. or maybe correct for sequencing errors, right. So, maybe if you are working with long read sequencing data as well as short read sequencing data, you can use the short read sequencing data to correct the long read data, right. So, that will reduce the error. So, you can take different approaches to address these issues. Okay, so what do we get after we have done this de novo assembly, right? So, whether we have used Wells approach or DBG approach or SCS approach, whatever the approach is, what do we get at the end? So, we get usually multiple contigs. Okay, as I mentioned, right, repeats remain in many cases, and even with DBG assembly, these repeats will persist because of the non-uniform coverage. So, we end up with these multiple contigs, and there are gaps between these contigs. Okay. And to cover this, we do something called scaffolding or gap closure. So, this is usually done between paired end using paired end reads, right. So, imagine you have these two contigs, right. So, this is contig 1 and this is contig 2, right, and in case you have some reads between these contigs, right. So, this paired end data, you can use those to actually this do the gap closure, or what we call as scaffolding. So, as you will go about this assembly process, you will see you can have these different levels of scaffolding. You can specify what level of scaffolding you want, etc. Okay. And here is a reference describing some scaffolding algorithm that can be used for this purpose. Okay, so, I will just now mention some of the tools that you can use for the assembly. So, we have discussed the algorithms, and these are implemented in different tools. Of course, they are based on some of these algorithms, but they take slightly different approach, right, some different implementations, right. And the early tools for assembly were the Celera. This is the assembler used for human genome assembly, again based on Wells approach, and then we also had the Nibbler, which was the 454 sequencing assembler. For assembly today, you have Velvet available. So, this is based on De Bruijn graph method that we discussed. Here is the link, and here is the reference paper that you can go through. Similarly, you have other tools. I am just mentioning some of them. I will not discuss because we have discussed the algorithms. The implementations might be slightly different. I will just give you references in case you want to read more and understand these tools better. You will find the links here. Okay, so, here is another tool called Hinge. This is Wells based approach, and here is the link and the reference paper. And similarly, you have now other sets of tools that are based on De Bruijn graph assembly. So, one is the SOAP de novo version 2, then you have SPA DES, again this is De Bruijn graph based assembly method. You have another one which is the IDBA. It actually has an interesting extension which can work with highly uneven coverage or depth, as you can see in the title of the paper. So, this is again a very interesting extension of the original tool, which is the IDBA. And finally, there are also other tools that actually can utilize other types of methods which we have not discussed, other types of algorithms or extensions of those algorithms. So, here is one which is called the SGA. This is a string graph based assembly, and here is the paper and the link to that. Now, once you have done the assembly, you need to assess the quality of the assembly, right, as we have done all throughout. Okay, we check quality of the read, we check quality of mapping, and here we want to check quality of assembly. We very briefly talked about this assembly quality when we are talking about transcriptome assembly. So, similarly here when you are doing genome assembly, you would also have to look for the quality. And one of the metrics that we defined earlier is called N50 metric. Okay. And the explanation for N50 is very simple. So, let us say N50 value is x, it means that at least half of all assembled contigs are of length x or higher. Okay. So, if x is let us say 5 kb, it will say, right, at least half of all assembled contigs will be greater than 5 kb. Okay. So, this is how we actually look at the data. So, and you want this value to be higher, that would mean you have managed to get close to the actual chromosome or genome. Okay. So, we also look at contig numbers, and we want these numbers to be small, right, as few as possible, and in ideal world we want these to be close to the chromosome number. Okay. And in addition, we also check for genomic coverage and genomic completeness. Okay, so how do you check this? So we have from other experiments, we have some idea about the genome size, number of chromosomes, etc. So we can utilize that information and see, right, how much of that genome size is covered by these reads that we have or the contigs that we have generated, right, how much of the full genome we have covered with the reads, and also the coverage, right, how many reads on average we see for each location of the genome. So these are very important parameters, and these actually have been implemented in different tools. Again, I will just simply mention these tools. Again, you can find the links, and you can also go through the papers, and you can utilize them for your research. Finally, the final step after you have checked the quality is something called genome annotation. Okay. So, you have derived a new reference sequence, right, but then it is simply just the basis, right, A, T, G, C, etc. So, what we need to do now is to add more information or add something, some association, or associate these bases to genes, exons, etc. Okay, and this process is called genome annotation. Okay, so there are two types of annotation that you can do. So the first is structural annotation. Okay, so this is about identifying structural elements of the genome, for example, genes, coding regions, exons, or introns. And this you can do through something called gene prediction, right. You can of course check closely related species or organisms and see whether there are some sequences that are resembling genes from those closely related species, right. So you can do this gene prediction, or you can also combine this analysis with transcriptome analysis, right. So the advantage is with transcriptome, you know which regions of the genome are transcribed, right. So you can then now start identifying the gene regions, the coding regions, exons and introns, right. Those we can do very easily if we also combine genome data with transcriptome data. The second type of annotation we do is called functional annotation. This is actually much harder, because what we want to do is we want to infer gene functions, right. So we have now identified some genes, but we want to infer gene function, and we have to do this through comparative analysis, because the experimental determination of function is very difficult, tedious and time consuming, right. So we would have to do this through comparative analysis with related genome, see whether similar genes exist in other organisms, and if we know the function of those genes in the related organisms, we can then say, okay, this gene also has similar sequence, so it is likely to perform the same function or similar function. Alternatively, you can also do domain predictions. So for specific functions, you have specific protein domains, and if you see presence of certain domains in certain genes, you might you might predict, right, okay, this gene is probably associated with this kind of function. Okay, so here are the references for this class, and to summarize, we have extensively discussed De Bruijn graph assembly. We have seen that it does not require any comparison for overlaps, right. So it builds this overlap graph or multi-graph using the k-1 merge in the data, and then we have seen, right, we can identify whether there is an Eulerian path, and this enables derivation of reference sequence. And of course, we also talked about exceptions, right. So different scenarios where we cannot find an Eulerian path, or where there could be multiple Eulerian paths. And we have talked about assembly quality assessment, because once we have done the assembly, we want to check how good the assembly is. Okay. The tools that we have described or the algorithms that we have described, as you can see, they are not perfect, right. There is still a lot of scope for improvement. And then finally, we talked about the genome annotation very briefly. This is for identification of structural and functional elements, and this actually follows after you have done the assembly, and this actually completes the whole process. Thank you very much.